This video will cover subtopic 3.3, meiosis. Here are a list of understandings, applications and skills, and guidance that will be covered in this video. So we'll start with human chromosome number and structure. So just recall that humans are diploid, so we have 2 of each type of chromosome, and our diploid number is 46. So we have 46 chromosomes in total. So in humans, the 46 chromosomes in somatic cells are not 46 different units, but 23 pairs of homologous chromosomes. So we have 23 different types of chromosomes, but we have 2 of each type of chromosomes. We have 46 chromosomes in total, 23 different types. So where do those 46 chromosomes come from to add up to 46? So 23 chromosomes you get from your mom, 23 chromosomes you get from your dad. So with those homologous chromosomes, each homologous chromosomes will carry information for those traits or for those genes. And just recall that each gene has a particular locus. The genes for specific traits will be found at the same loci on each of the homologs, but each homolog may or may not have different alleles. And just recall that alleles are the different alleles are variations of those genes. So looking at chromosome structure, so there are 2 forms of chromosomes. So they're unduplicated and duplicated or replicated. So unduplicated chromosomes would just be referred to as a chromatid. And this is unreplicated DNA. So when the DNA gets replicated now, basically you're doubling the chromosome that's present, and why you get this X structure. So this top part of the chromosome would be replicated, the bottom part of the chromosome would be replicated, and you get this X structure. And so these duplicated chromosomes contain the identical sister chromatids. So this would be 1/2 of the chromosome, this would be a sister chromatid, and the other half would consist of the other sister chromatid. Now, chromosomes from an organism can be used to make a karyogram, and a karyogram can be used to determine the karyotype. So this image here shows the unreplicated chromatids all lined up from biggest to smallest with the sex chromosomes at the end. The following karyogram shows the replicated chromatids in that typical chromosome structure. So meiosis comes into play during sexual reproduction and production of gametes. So gametes include cells that would be sperm and egg. Sperm and egg are haploid cells, meaning they have 1 of each type of chromosome. So in humans, they would have an N number of 23. Meiosis is the process that will produce these gametes. And just when these gametes fuse together, so when you have a sperm cell that fertilizes an egg cell, it produces that zygote. So if the sperm is N, meaning it has 23 chromosomes, the egg is N, meaning it has 23 chromosomes, those 2 chromosomes, 2 sets of chromosomes will produce or will combine to produce a diploid organism, that is 2N. And so that process of fertilization is what will produce that diploid zygote. So fertilization really doubles the number of chromosomes. So the discovery of meiosis was done with improved microscopes, and it was discovered that some dyes stained the nucleus of the cell. These dyes revealed thread-like structures in dividing nuclei and were named chromosomes. German biologists carried out observations of these dividing nuclei that revealed how mitosis and meiosis occur. So your task is to do a little bit of reading on page 160, make some notes as to how scientists did this or made this discovery. So getting into meiosis, or this process of producing haploid gametes, there are two big key outcomes. The first key outcome of meiosis is genetic reduction. So it produces daughter cells with half the number of chromosomes of the parent cell. So basically, you start with a diploid cell, and you're going to a haploid cell. So that is the big key, one of the big key outcomes. The other key outcome in meiosis is genetic recombination. So the products, those gametes, have different combinations of alleles, and as a result, offspring are going to be genetically different from one another and their parents. So think about your siblings and the fact that, you may not look exactly alike, you may be different. This is the result of genetic recombination. So having a look at meiosis, a big overview of meiosis. So we'll start in general with interphase here, and we start with our unreplicated chromosomes. Note there is one chromosome here and a homologous chromosome with it. So these, this blue chromosome and the red chromosome indicate, that they're homologous in this case. So they get replicated to produce these sister chromatids, and from here the cell will enter into meiosis 1. Now at the end of meiosis 1, the big thing that happens is that the homologous chromosomes separate. So when anaphase occurs, the homologous chromosomes get separated. The sister chromatids remain intact at the end of meiosis 1. So here's where you get your genetic reduction. So a cell that started out as 2N now goes down to a haploid cell, or N, at the end of meiosis 1. In meiosis 2, this is where you now get separation of the sister chromatids. So these sister chromatids now separate into separate cells. The end result at meiosis 2 is that you end up with 4 haploid cells. Going through each of the different steps in meiosis, it all starts with interphase. So in interphase, the chromatin has replicated. For each chromosome, the result is 2 genetically identical sister chromatids, which are attached at the centromere. So basically, those 46 chromatids will duplicate to 92 chromatids. Now, at the start of meiosis 1, the key events here is that you'll get those homologous chromosomes that will separate, and the cells will initially have one copy of each chromosome, one from mom, one from dad. So the cell will start out as being diploid. And the cells separate such that daughter cells have either one copy of each chromosome from mom or dad as they become haploid. So you get that 2N diploid cell that becomes haploid. In prophase 1, we'll have a look at the cell parts involved. So there are the centrioles that will start to move to the opposite poles of the cells. You get microtubules that form between the centrioles. The nuclear envelope will start to disappear, and the spindle fibers will attach to the centromere of each homologous pair of sister chromatids, meaning that you'll only get a spindle fiber, one spindle fiber that just attaches to each homologous chromosome. So the homologous chromosomes will separate in this manner. Having a look at the chromosomes in prophase 1, so the chromosomes will condense to form those sister chromatids. The homologs, which consists of 2 sets of sister chromatids, pair up to form what is called a tetrad. So when you have a pair of homologous chromosomes, they're called a tetrad because you basically have 4 sister chromatids together. The chromatids of homologous chromosomes will be crossed at what is called a chiasmata, so you can get an exchange of chromosome between homologous chromosomes at this area called a chiasmata. So basically, like you'll have the chromosomes swapping identical parts. So like some of the blue chromosome now will end up with the red chromosome, and some of the red chromosome will end up with the blue chromosome. And ultimately, this will help increase genetic diversity. From here, you go to metaphase 1. This is where the bivalents, or the tetrads, and we'll continue to refer to them as tetrads, line up along the metaphase plate. So they'll line up along the center of the cell, and the orientation of the paternal and maternal chromosomes is going to be random and independent, meaning that you don't get all of the maternal chromosomes on one side and all of the paternal chromosomes on one side. How they orient themselves, or which pole they orient themselves to, is going to be random. In anaphase 1, this is where the homologous chromosomes will separate and move to opposite poles of the cell. Note that the homologous chromosomes separate, they do not split when they move to the opposite poles of the cell. The sister chromatids are held together, and the centromeres do not split. At this point, this is where the cell goes from being diploid to haploid. So here we have our homologous chromosomes. The chromosomes now are moving to opposite poles of the cell. So when the telophase and cytokinesis occur, the newly formed cells will contain one of each type of chromosome. In telophase 1 and cytokinesis, the homologous chromosomes will uncoil and the spindle fibers will disappear. A cleavage furrow will form and cause the cytoplasm to divide, and the nuclear membrane will start to reform. The result of this is two haploid cells. In meiosis 2, the big events that occur is that you will get separation of these sister chromatids, and at the end you will get 4 haploid daughter cells that result containing a single chromosome. So these cells will start haploid and will end haploid. In prophase 2, the centrioles will now move to the opposite poles, so centrioles start to move to the opposite poles. Spindle microtubules will form between the centrioles, the nuclear envelope will disappear, and the sister chromatids will condense. So the same series of events as in prophase 1 occur in prophase 2, just now this time we have 2 cells where this is occurring, whereas in prophase 1 it was just 1 cell. In metaphase 2, the sister chromatids are going to line up along the metaphase plate, and spindle fibers will attach to the centromere of the sister chromatids. So here the spindle fibers will attach, you'll get 2 spindle fibers attaching to 1 chromosome, or a spindle fiber attaching to each sister chromatid. So in metaphase 1, it was a spindle fiber attaching to a chromosome, and a spindle fiber attaching to each homologous chromosome, and that was what separated the homologous chromosomes. In metaphase 2, we're going to separate these sister chromatids, so you need a spindle fiber attaching to each sister chromatid this time. During anaphase 2, the centromeres of the sister chromatids will separate. Notice the word separate, they do not split. So they separate and will move to opposite poles of the cell. So now the resulting cell is going to end up with 1 chromatid of each chromosome. And our haploid cell is going to remain as haploid. In telophase 2 and cytokinesis, nuclei form at opposite poles. The nuclei will reform at opposite poles of the cell, and cytokinesis occurs. So you get the cleavage furrow that forms, nuclear membrane that reappears, the chromatid will uncoil, and so the result now is that we get 4 haploid daughter cells. We have 1, 2, 3, 4, and if meiosis is occurring in a male, the result is 4 sperm cells. If it's a female, we do not get 4 eggs that are produced. We only get 1 egg. So we get 1 big cell, and then we get 3 smaller little blebs that come off of that as meiosis occurs, which are referred to as polar bodies. So these polar bodies have a very small amount of cytoplasm with the unwanted genetic information, or those unwanted chromosomes. So you get these 3 polar bodies, which result due to unequal cytokinesis. Now, meiosis is also important for genetic recombination. So meiosis will result in genetically distinct haploid gametes. And this process occurs through a couple different ways, through crossing over or recombination. And this is the exchange of material, of chromosomal material between the maternal and paternal chromosomes. And you can get genetically distinct haploid gametes by independent assortment. This is the creation of gametes that carry different combinations of maternal and paternal chromosomes. So in this process of crossing over, this occurs during prophase 1 and during metaphase as well, during metaphase 1 as well. And it will occur when the homologous chromosomes line up, and can occur at several points along non-sister chromatids. So basically, once these homologous chromosomes pair up, once you get these tetrads that form, you can get sections of these chromosomes that will switch spots. And it will be identical spots. Remember, the gene sequence on these chromosomes is going to be identical, so you can get switching of these chromosomes and maintain the same genetic sequence. So you can get that crossing over at that chiasmata, where a portion of, in this case, the red chromosome will now join up with the orange chromosome, and a portion of the orange chromosome now joins the red chromosome. And so now the result is that you get chromosomes that contain genes from maternal and paternal origin. So this chromosome that was originally paternal and the other chromosome that is maternal, the end result is that you get a mix of paternal and maternal chromosomes. In independent assortment, this occurs during metaphase 1. Each pair of maternal chromosomes is oriented towards one pole, and paternal chromosomes is oriented towards the other pole. Depending on how the chromosomes line up, it can produce different combinations of gametes. So in this diagram here, we have our chromosomes. So we have, chromosomes from dad, chromosomes from mom, and depending on how they line up in terms of what pole they're pointing at, you can get different combinations. So this diagram shows the other possible combination in terms of how these chromosomes could line up. So here we have, the big chromosome from dad pointing to one pole, and the small chromosome from mom pointing towards a similar pole. So when anaphase 1 occurs, you'll get the big chromosome from dad and the small chromosome from mom together in one gamete. In the other and the other gamete, in this case, will have the big chromosome from mom and the small chromosome from dad. Now, the other possible arrangement is that all the chromosomes, in this case from mom, point or will go towards one pole, all the chromosomes from dad will go to the other pole. And so one gamete will contain chromosomes from mom, one gamete will contain chromosomes from mom. And so when meiosis 2 occurs, you end up with gametes that contain just chromosomes from mom and 2 gametes that contain chromosomes just from dad. Whereas in the first example, the gametes that are produced are a blend of chromosomes from mom and dad. Now, without recombination, without that crossing over, you can basically end up with 2 to the n distinct gametes. So n being the number of chromosomes or types of chromosomes that an organism has. So in the case of humans, because we have 23 different types of chromosomes, we can have 2 to the 23 types of gametes, possible gametes that are produced. And this has to do with the various ways in which the chromosomes can orient themselves and the various ways they can line up during metaphase 1. So there's a lot of different possibilities there in humans. Now, there can be errors caused by changes in chromosome number, and this can happen during meiosis 1 or meiosis 2. Now, these changes in chromosome number result from non-disjunction. This is where homologous pairs or sister chromatids do not separate as they should in meiosis. So for the homologous pairs, this will occur in anaphase 1. So those homologous chromosomes do not separate to opposite points. And for the sister chromatids, it would be in anaphase 2, where they do not separate. So the result is gametes with too many or too few chromosomes. So in the diagram here in meiosis 1, here we have homologous chromosomes that have not separated. So both of these chromosomes have gone towards the same pole of the cell. And so during meiosis 2, when you get separation of those sister chromatids, you end up with two gametes with too many chromosomes in them. So they all have one additional chromosome. So they're basically trisomy. And here we have gametes that have lost a chromosome, so they are missing a chromosome. In meiosis 2, non-disjunction can occur in anaphase 2, where sister chromatids do not separate. So in this case, both sister chromatids have moved to the same pole, which results in one gamete with extra chromosome in it, one gamete that is missing a chromosome. Because the homologous chromosomes separated properly at the start, the second daughter cell that resulted at the end of meiosis 1, and with proper separation of the sister chromatids, these gametes here have the correct number of chromatids at the end of meiosis 2. So meiosis can cause an increase in chromosome number or a decrease in chromosome number in some gametes. Now, if fertilization occurs with these gametes that have extra chromosomes or a loss of chromosome, it can result in genetic conditions. So there is monosomy, where you have the loss of a chromosome, and you have trisomy, where you have the gain of that extra chromosome. And the most common trisomy is trisomy 21, which is referred to as Down syndrome. And this is an extra copy of chromosome 21. Now, when determining the karyotype of an individual, when examining those chromosomes to create a karyotype, and determining if a fetus or a baby is going to have a normal chromosome pattern or an abnormal chromosome pattern, there's a few different ways in which you would obtain cells from a fetus to do this karyotyping. Now, these tests are invasive and they do carry risks with them. They do involve collecting DNA samples of the fetus. So there's two ways in which this can be done. One through a process called amniocentesis and another called CVS, or chorionic villus sampling. Now, in amniocentesis, a needle is used to withdraw a sample of amniotic fluid from the amniotic sac. So a needle is placed in through the abdomen into the amniotic sac with guidance of an ultrasound scanner to make sure the baby is not injured, or that fetus is not injured. A sample of fluid is retrieved containing those fetal cells. Using those fetal cells, you can examine the chromosomes of the developing fetus. Now, there are risks of a miscarriage of about 1%, and generally this is done at around the 14th week of gestation. With CVS, or the chorionic villus sampling, this is done earlier on in the pregnancy and carries a higher miscarriage rate. So this is typically done at around the 9th week. So a catheter is used to obtain a sample of placenta and is inserted in through the vagina up to the uterus, where a sample of placenta is obtained, again, with the guidance of an ultrasound scanner. And those cells that are obtained are then used to obtain the chromosomes that are used to create that karyotype. Now, parental age plays a role in non-disjunction, and it's the mom's age that actually influences the chances of non-disjunction occurring. So the following graph shows maternal age as well as the percent risk. The blue line represents the risk for trisomy 21, Down syndrome, and the red line represents risk for all trisomies, for all the different types of trisomies, including trisomy 21. So at very young age, in 20s and even very early 30s, the risk for trisomies are very low. But once that maternal age hits at around 35, 36, the risk for trisomies start to increase. And as that maternal age gets closer to 40 and hits 40, that risk increases significantly. So in late 40s, there is a very high risk of trisomy 21 and a very high risk, 1 in 8 chances of any type of trisomy occurring very late on with that maternal age. So this is the end of the video for subtopic 3.3, meiosis.